Very good morning and welcome to Unacademy. This is Abhijit and today we are going to discuss about the IMP questions from biology for competitive examinations. Okay, but before starting the lecture, it is very necessary to know about the few things. Okay, so the first thing is this channel Crack Maharashtra CET. Okay, this channel is running as per the new syllabus, so it is necessary to follow for the 2021 examination of CET. Okay, timing of the lecture is in the front of you, so as per the timing, all the lectures were conducted. So before starting the lecture, it is very necessary to know about me. Uh, My name is Abhijit Madkar. I have more than 6 years of experience in the field of biology and also crack net CSIR from life science. So guys, we are here on the India's largest platform, the Unacademy. So this is app of Unacademy. This app will give you the thousands of lectures from the thousands of educators. Okay, so the link of this app is given below in the description of this video, so you can download this app from there. And this is Telegram app. If you do not have a Telegram app, the link is given below in the description of the video. And if you have a Telegram app, just go through the link given below in the description for the channel. Okay, so if you join the channel will provide you the notes, MCQs, and the links. Okay, so these three things are available on this channel, and you can attend the live lectures from these links, so you will have the chance of interaction with your educators. Okay, so when you are live with any lecture, so try to ask the questions, try to ask the problems if you have any. Okay, so guys, it is a good thing. तुम्हाला सगळ्यांना हा चान्स भेटलेला आहे एज्युकेटर सोबत इंटरॅक्ट होण्याचा. हा चान्स इतके दिवस नव्हता आपल्याला. आपल्याला सगळ्या गोष्टी व्हर्च्युअल असायच्या किंवा टू वे कम्युनिकेशन होत नसायचा. आज चान्स आहे, तुम्ही क्वेश्चन विचारत चला. ज्यावेळेस पण कोणता एज्युकेटर तुम्हाला लाईव्ह असेल, फिजिक्स असो, केमिस्ट्री असो, मॅथ असो, बायोलॉजी असो, तुमच्या विषयाचा, तर त्यावेळेस तुम्ही त्यांना क्वेश्चन्स विचारा आणि टू वे कम्युनिकेशन करण्याचा प्रयत्न करा. So guys, these are the few subscriptions available for you. So if you see the subscription of a 12 months, so the fee is 14,850 rupees, and for 24 months it's 21,600 rupees. If you use this capital Abhijit as a subscription code, it will again give you the 10% discount in a fee. Okay, या फी मध्ये 10% डिस्काउंट भेटणार आहे. म्हणजे ही फीस तुम्हाला 13,000च्या आसपास आणि ही फीस तुम्हाला फक्त 20,000च्या आसपास किंवा 18,000च्या आसपास तुम्हाला भेटणार आहे. ठीक आहे, यामध्ये 2,160 रुपये तुम्हाला डिस्काउंट भेटणार आहे आणि यामध्ये 1,485 रुपये. ठीक आहे, म्हणजे तुम्हाला फी फार कमी आहे आणि यामध्ये सगळे लेक्चर्स, ऑल सब्जेक्ट्स, देन एमसीक्यूज प्रॅक्टिस, एमसीक्यूज पेपर पण तुम्हाला इथं आहेत, त्याचा अनालिसिस आहे. अनालिसिस मध्ये तुम्हाला तुमच्या अभ्यासाची डिरेक्शन फाइंड करता येते. त्यानंतर तुम्हाला यामध्ये डेडिकेटेड डाउट सॉल्विंग सेशन सुद्धा अवेलेबल आहेत. त्यानंतर तुम्ही तुमच्या फेवरेट एज्युकेटर सोबत इंटरॅक्ट होऊ शकता. ऑफलाईन फॅसिलिटी, या सगळ्या गोष्टी तुम्हाला एका सबस्क्रिप्शन मध्ये अवेलेबल आहेत. सो यू कॅन से द सिंगल सबस्क्रिप्शन अँड द अनलिमिटेड यूज ऑफ ॲप. So when you are taking this subscription, so make sure that it will not going to end before the examination. Okay, so let's start with our session, guys. The IMP questions from biology for CET examination. Okay, so the first question is in front of you. Bond formed between the first phosphate and the adenosine in a ATP is nitrophosphate bond, adenophosphate bond, phosphoanhydride bond, and the phosphoester bond. Okay, so guys, let's see the actually where this bond present. Okay, so for example, this is your ATP. Okay, here is adenine, and the three phosphates are connected here, for example. Okay, so your let's see, This is adenosine. Adenosine means, okay, and the 3 phosphate, for example, here we can draw like this now. So this bond between the adenosine and the phosphate is, so this bond, if you say, it is the phosphoester bond. Okay, so if you see in the DNA, okay, so adenine is, for example, here A, okay, your phosphate bond is here. So this is again the phosphoester bond. When you say this bond attached to the fifth carbon atom of ribose sugar here, so now you can say this bond is a phosphodiester because two ester bonds are there, and that's why diester here is, for example, any ATGC, whatever it is. Okay, so here present, and in the ATP, it is also the like the phosphate bond is here, triphosphate. Okay, so this is sugar and the adenine is here. So this bond between the sugar, adenine, and the phosphate is called as phosphoester bond. Okay, so this is phosphoester bond, uh, which is explained into the question. Okay, so make sure that, guys, when this bond is present at the both side, so we mostly call it phosphodiester bond. Okay, डायस्टर दोन्ही साईडला असेल तर, एका साईडला असेल तर ईस्टर बॉन्ड. द युज्युअल शेप ऑफ ग्रोथ कर्व. ग्रोथ कर्व बद्दल आपल्याला माहिती आहे की ग्रोथ कर्व मोस्टली आपण काढताना जसा काढतो त्याला, की पहिल्यांदा फॉर एक्झाम्पल इथे 0 सेल्स आहेत, काही सेल्स तुम्ही टाकल्या की तो इथून सुरू होतो. ठीक आहे, सेल्स टाकल्या की पहिल्यांदा त्या एन्व्हायरमेंट सोबत त्यांना कोप अप करायला वेळ लागतो, and that phase, if you see, the time is going to be a increase, but the number of cell is not going to be a very fast increase in a very fast. Okay, so here is a number of cell and here is a time. Okay, so the first graph seems like this, and then it moves like the proportional with n and t. Okay, and finally it become like a flat. Okay, but we draw it like this, and again come back to the dead phase. Okay, so now from here, if you see, the graph is going to be a decline, so it is called as a dead phase. But mostly we draw this graph like this, and again flat, so it's a sigmoid graph, or sometimes we call it a S phase, oh sorry, S shape graph. Okay, shape, if you see, it's a sigmoid, or you can say the S shape graph. Okay, this is called as exponential phase, or this is called as the stationary phase. Here is exponential growth. Okay, it's a growth phase, and here the stationary phase comes where the secondary metabolites uh, started making here. Okay, secondary metabolites इथे सुरू होतात बनवायला, आणि हा growth phase आहे where the number of cell going to be increased very fast as compared to the time, because it's exponential growth. For example, here is a 4 cell. After one division we will have the 8, then 16, then 32. Each and every division going to be a increase amount of a cell, like a double, 128, 256, 520, uh, 512, 1024. So it is going to be a increase, not increase, but the going to be a double. That's okay, beta. When you got like I am live, so that time just just join it. Okay, so don't worry. Sometime it happens. Okay, so this is the shape. It's not linear and it's not inverted bell shape. So what is meant by inverted bell shape, and the which graph shows inverted bell shape. Okay, so we will discuss it into the environment. Okay, the ultimate cause of a water movement against the gravity in a plant is, the ultimate cause of a water movement against the gravity. So if you say, for example, this is a ground. This is the your plant. It's a tap root system. Okay, so I'm going to draw a dicot like a plant here, for example mango tree. So I just draw it a taproot system. Okay, so please keep in mind that the taproot system and the adventitious root system, monocot and dicot. Adventitious in monocot and the taproot system in a dicot. Okay, so the branches and the other things are there in a plant, and this is your plant, for example. Okay, so for the sake of understanding we are going to try it. Okay, so don't look like a technically correct or the incorrect things. Okay, so this is a plant, for example. We know it is filled with the water. Okay, so the water is coming from here, enter into the root, and finally moves toward the upper portion of a plant. Okay, when it come the at the surface of a plant where the sunlight is striking. Okay, so here the sunlight is striking on a plant, and the plant is going to lose water in the form of a vapor. Okay, so the S2O in the gaseous form or the vapor form coming out of the plant. So this is called as the uh, evaporation of water in your language, but if you see as per the a concept in a plant, it's called as a transpiration. Okay, so it's a ट्रान्सपिरेशन म्हणजे वॉटर वेपरच्या फॉर्म मध्ये बाहेर निघणं. आता ट्रान्सपिरेशन मधून जे वॉटर बाहेर निघतं ना, इट creates a pool here, and this pool is responsible for the pulling of water from the bottom of the plant. Okay, इथे पूल तयार होतो एक, आणि तो पूल काय करतो, पूल म्हणजे पूल प्रेशर आहे, सक करतो तो पाण्याला खालून वरती ओढतो, आणि हा जो पाण्याला खालून वरती ओढणारा जो प्रेशर आहे, तर त्या प्रेशरला आपण ट्रान्सपिरेशन पूल म्हणतो. So the movement of water against the gravity in a plant is due to the transpiration pull. Okay, so the osmosis and the interaction between the same molecule, interaction between the different molecule, ज्याला तुम्ही adherent force किंवा त्याला तुम्ही cohesion फोर्स किंवा इंटरॅक्शन हे जे शब्द वापरताय सगळ्या गोष्टी तिथे आहेत, पण ट्रान्सपिरेशन पूल एक मेन कॉन्सेप्ट आहे की जो वापरला जातो. इम्बिबिशन सुद्धा तिथे असते. इम्बिबिशन म्हणजे ज्यावेळेस पण dead part plants are, but having the hydrophilic material like a cell wall and pectin, they are able to suck the water, they are able to hold the water, and when water is moving from the a dead part of a plant like a cell wall and the gap between the cell two cells, so this pool or this the movement of water or this absorption of water is called as imbibition. Okay, जसं की पावसाळ्यामध्ये आपल्या घराचा दरवाजा फुगतो की नाही ओला झाल्यावरती, तर तो इम्बिबिशन मुळे फुगतो, त्याला इम्बिबिशन म्हणतात. ठीक आहे सर. एमएसटी सीईटी 2021 की ऑफिशियल अनाउन्समेंट कब होगी? बर, झाल्यावरती सांगेल मी तुम्हाला. गुड मॉर्निंग. ओके प्रेरणा, व्हेरी गुड बेटा, इट्स करेक्ट, ऑप्शन सी. ओके, so let's see the next one. Jaundice is a disorder of. Okay, so jaundice is a disorder of. So is there anyone? See, बर, digestive system. Okay, so we got one answer, digestive system. We will discuss it definitely. Any other? Anyone please? Okay, so jaundice is actually the malfunctioning of liver. Okay, so for example this is liver. We will have the gallbladder here. Okay, this pipe is moving toward the small intestine. Okay, small intestine, And what happen inside the liver? So liver is having a Kupper cell inside. Okay, Kupper cell. Okay, these Kupper cells are the macrophages type cells. They are involved in a killing of RBC. Killing of RBC. RBC काय आहे रे? RBC red blood cell. Okay, erythrocytes. So it is made up of only hemoglobin. So it is filled with hemoglobin. There is no any organelle present inside of RBC. We know that it's a dead cell having only hemoglobin. If you see the hemoglobin, ferrous is between the one ring. This ring is called as porphyryl ring. Okay, so it's a porphyryl ring. शॉर्टकट वापरतो पेपरला लिहू नका असले. आणि हा फेरस आयन. ओके, आणि याच्या बाजूला तुम्हाला इथे एक प्रोटीन पाहायला भेटेल, असं प्रोटीन. त्यामुळे जर तुम्ही पाहिलं तर हिमोग्लोबिन इज मेड अप ऑफ प्रोटीन, porphyryl ring, and the last thing is a ferrous. Okay, so when the RBC is a kill, का RBC मारायची? तिची लाईफ कंप्लीट झालेली असते. ओके, RBC लाईफ ऑफ RBC is 120 days. 120 days. ओके, लाईफ स्पॅन कंप्लीट झाला की तिची ऑक्सिजन कॅरिंग कॅपॅसिटी कमी होते, तिला मारून टाकलं जातं. किंवा कधी मध्ये एखाद्या कार्बन मोनॉक्साईडने किंवा दुसऱ्या कोणत्याही केमिकलने तुम्ही काही इनहेल केलं तर त्याने आरबीसी डेड झाली किंवा व्यवस्थित काम करत नाही. सो दॅट टाइम द रिप्लेसमेंट ऑफ आरबीसी इज डन बाय युअर liver. And the second thing, spleen. Okay, so these two things or these two organs are considered as the ग्रेव्हयार्ड ऑफ आरबीसी. ग्रेव्हयार्ड म्हणजे मशानभूमी. ओके, आरबीसी ची मशानभूमी म्हणतो आपण यांना, ग्रेव्हयार्ड म्हणतो आपण या दोन ऑर्गन्सला, मोस्टली स्प्लीन आणि त्यानंतर लिव्हर. ओके, स्प्लीनचा नंबर अधिक यायचा. आता ही आरबीसी जर पोडली किंवा डेड तुम्ही कट करून आरबीसी ला आता रिप्लेस करायचंय. प्रोटीन कट करणार, प्रोटीन एका बाजूला काढले. प्रोटीन इज फायनली कन्वर्टेड इनटू द अमायनो ऍसिड अँड दिस अमायनो ऍसिड कॅन बी यूज्ड बाय अ बॉडी फॉर द डिफरंट पर्पजेस. फॉर एक्झाम्पल एनर्जी बनवायची, त्यातला नायट्रोजन काढून टाकला बाय दी डिअमिनेशन प्रोसेस. ओके, नायट्रोजन काढायचं म्हणजे NH3 कंप्लीट निघतो, अमोनिया बाहेर पडतो. डिअमिनेशन प्रोसेस म्हणतो त्याला, डिनायट्रिफिकेशन वगैरे म्हणून चुकवायचे नाही पेपरला आन्सर. ओके, अमायनो ऍसिड मधनं काय केलं जातं? डिनायट्रिफिकेशनने NH3 काढला जातो. हा इथेच होतं लिव्हर मध्येच. आणि जो राहिला आहे त्याला कार्बन स्केलेटन म्हणूया आपण. कार्बन स्केलेटन यातला जर C2 असेल तर हा एंट्री करतो लिंक रिएक्शन मध्ये. लिंक रिएक्शन म्हणजे मायटोकॉन्ड्रियाच्या बॉर्डर वरती होते आणि लिंक रिएक्शन नंतर क्रेब सायकल, आणि क्रेब सायकल नंतर इलेक्ट्रॉन ट्रान्सपोर्ट सिस्टम, आणि नंतर आपल्याला एटीपी मिळतात. हा एक यूज असतो. किंवा कार्बन स्केलेटन वेगवेगळ्या कारणासाठी यूज केले जातात. समटाइम दे आर डायरेक्टली यूज्ड इनटू द क्रेब सायकल लाईक फिलिंग रिएक्शन्स. फिलिंग रिएक्शन्स. ओके, म्हणजे या अमायनो ऍसिडचा तुम्हाला भरपूर वेगवेगळा यूज आहे. ठीक आहे, राहिलं काय तुमच्याकडे आता? पर्परिल रिंग आणि फेरस. फेरस स्टोअर केला जातो लिव्हर मध्येच, इथे स्टोअर होतो आणि स्टोअर पेराटिनच्या फॉर्म मध्ये होतो. म्हणजे पेराटिन प्रोटीन आहे त्याच्यावरती स्टोअर केला जातो फेरस. फेरस आपण फेकून देत नाही. प्रत्येक वेळेस आरबीसी पोडली आणि जर फेरस एक्सक्रीट केले बॉडीच्या बाहेर तर एवढा फेरस आपल्याला रोज रोज मिळणं शक्य नाहीये. ठीक आहे, त्यामुळे आपण फेकत नाही, त्याला स्टोअर करून ठेवतो. राहिली पर्परिल रिंग. ही पर्परिल रिंग ठेवता येत नाही बॉडीमध्ये, हिला कट केलं जातं. ही रिंग जर तुम्ही पाहिली तर फॉर एक्झाम्पल अशा एक 5-6 रिंग आहेत अशा एकमेकांना अटॅच केलेल्या. 4 रिंगला मध्ये वेगवेगळ्या परत रिंग आहेत, पण मेन 4 रिंग्स आहेत म्हणून मी ते 4 एरो दाखवलेत असे. ओके, फॉर एक्झाम्पल 1 2 3 आणि 4. ओके, या रिंग तोडल्या जातात आणि या तोडल्यानंतर 2 मध्ये डिवाइड होतात. ठीक आहे, मग ही पर्परिल रिंग तोडली की 2 मध्ये डिवाइड होते. एकाचं नाव आहे बिलिरुबिन आणि एकाचं नाव आहे बिलिवर्डिन. ओके, बिलिरुबिन आणि बिलिवर्डिन. हा बिलिरुबिन आणि बिलिवर्डिन येलो आणि ग्रीन कलरचे कंपाऊंड आहेत. दे आर शोविंग the alkaline nature. Okay, and due to that these compounds are going to be a store in the gallbladder मध्ये या दोघांना स्टोअर केलं जातं असेल बिलिरुबिन आणि बिलिवर्डिन, आणि यांना इथून स्मॉल इंटेस्टाइन मध्ये टाकलं जातं. वेअर इन स्मॉल इंटेस्टाइन दे न्यूट्रलाइज acids from stomach. स्टमक मधनं येणाऱ्या ऍसिडला न्यूट्रलाइज करतात. ठीक आहे, जर लिव्हर मध्ये काही डॅमेज झाला किंवा लिव्हर इज गोइंग टू बी अ माल फंक्शन, सो दॅट टाइम वी नो लिव्हर इज हॅव्हिंग दी वन सप्लाय ऑफ वेन So this is a vena cava. Okay, vena cava, inferior vena cava, moving toward the हार्ट. लिव्हरचं जे काय फंक्शन आहे तर याच्यामध्ये अशा ब्लड पाईप्स एकत्र केलेले आहे आणि ब्लड पाईप परत एकत्र येतात आणि एका ठिकाणी येऊन या वेना केव्हामध्ये जातात. मग हे सगळं मटेरियल ते देतो वेना केव्हामध्ये टाकून आणि मटेरियल तुमच्या बॉडीमध्ये फिरतं. कोण? हे दोघ जण, येलो आणि ब्ल्यू कलर, सॉरी येलो आणि ग्रीन कलर वाले कॉम्पोनंट, दे आर नाउ मूविंग इनटू द बॉडी अँड दे जस्ट सेटल डाऊन इन द व्हाईट पार्ट ऑफ द बॉडी. व्हाईट पार्ट मध्ये दोन गोष्टी येतात, एक येतो डोळ्याचा पांढरा भाग, आय, द व्हाईट पार्ट ऑफ आय, अँड सेकंड वन इज अ नेल. ओके, so in this two part, this yellow color material is going to be a settle and we called it a jaundice. So in a jaundice, the body is not going to, mostly your liver is not going to function very well, and because of it results into the jaundice. So there are few causative agents for jaundice like hepatitis, like viruses, and bacteria is also. Jaundice may be, I mean the jaundice is actually the symptom of having damage in a liver. Okay, jaundice का आहे? सिम्टम आहे, लिव्हर मध्ये डॅमेज आहे याचा एक सिम्टम आहे जॉन्डिस. आणि जर जॉन्डिस अशा पद्धतीने होत असेल तर गरजेचं नाही की याला एखादा पॉझिटिव्ह एजंट पाहिजे. जर जोरात उजव्या बाजूला लिव्हर असतो आपला या अब्डॉमिनल कॅव्हिटी मध्ये, जर जोराचा फटका त्याला लागला किंवा जोराचा टेबलचा कॉर्नर वगैरे असं काही चालताना लागलं आणि लिव्हरला जर सूज आली किंवा डॅमेज झालं तरी पण जॉन्डिस होऊ शकतो. ठीक आहे, तर जॉन्डिस म्हणजे कावीळ म्हणतो आपण त्याला मराठीमध्ये. तर ही लिव्हरच्या माल फंक्शनिंग मुळे होते. अँड लिव्हर वी नो इट्स a part of a digestive system, or you can say the jaundice is a disorder of a digestive system because the one thing is going to be hamper more is your digestive system because of the uh, low amount of release or the release of a low a juice, that is the bile juice from the liver, is result into the acidity into the small intestine. Okay, न्यूट्रलायझेशन व्यवस्थित होत नाही. चलो नेक्स्ट. प्लस फॅट्स पण डायजेस्ट केले जात नाहीत कारण फॅट्स डिझॉल्व करून आपण ज्याला इमल्सिफिकेशन म्हणतो तर ती इमल्सिफिकेशन होत नाही. द फायनल इलेक्ट्रॉन acceptor in electron transport chain in a respiration is, okay, so the final electron acceptor in your electron transport chain, for example this is a complex 1. So guys, please answer this question. This is complex 2, 3, 4, and the complex 5 is nothing but your lollipop complex. Okay, so this is complex 1, pump. Complex 3, pump. This is 2, it's a 3, pump. And the 4, it is also going to be pump, the thing. Okay, be oxygen, very good beta. So the electron from the NADH is given to the NAD plus and convert itself, it is oxidized form, and this is a reduced form. Okay, this is oxidized and reduced form. Finally it is given to the EMN sorry NAD, it convert into the oxidized form, and the electron is now by the coenzyme Q, we just take electron and we call it a QS2, and it moves toward the complex 3. Then electron is moving toward the complex 4, and after the complex 4 give this electron to the oxygen, which convert itself into the S2O, and this is the your final electron acceptor. You can say the strong acceptor, strong acceptor of electron. Okay, electron's a strong acceptor आहे, and your strong donor is NADH, and the one more donor is here FAD. So FAD is also the donor of electron. Okay, हे लिहून ठेवायचे, परत परत विचारले जातात. Very good beta, your answer is correct. Oxygen, your final acceptor is oxygen. Okay, and this is your 80 page. The plus chemosmatic theory आहे ही जी chemosmatic theory आहे पीटर मिशेल ने एक्सप्लेन केलेली आहे. सायंटिस्टचं नाव लक्षात ठेवा, पीटर मिशेल. ओके, so H plus is now again moving toward the matrix. This is a matrix part, and it is having its energy this energy is dissipated, and this dissipated energy is grasped by these ATPase or the one F naught complex, and we convert it into the ATP by taking ADP plus PI. Okay. So this is the final this is the complete overview of your um, electron transport system in a mitochondria. It is present in a inner membrane of a mitochondria. The root concentrate minerals by, okay, so for example, this is a root. Okay, this is plant, so not going to complete the diagram. And this is, for example, the roots. Okay, so so for example, a root want to absorb the water. So due to absorb the water, it is necessary to make the concentration gradient here. So it made the more amount of solute inside the root as compared to the outside. Okay, so due to the more solute, it causes the diffusion, generation of diffusion pressure, and due to the semi-permeability, this membrane is not going to allow this solute to come out of the cell, and that's why the water present into the soil, in a soil, is enter into the root to minimize this pressure, and it is pulled toward the apex of a plant by transpiration pull. So it always required ATP, which convert into the ADP. Okay, so the requirement of ADP for the transport is called as active transport. Okay, so this is active transport. The root concentrate minerals by the active transport process, and it is happen most of the time or mostly in summer days. Okay, so if you see in rainy days, there is plenty of water, so plant don't want to take more work or the more hard things. For that it is easy to absorb, but in summer days there is a very low concentration of water present inside of a soil, and there is also the capacity of a soil to hold the water and to lose the water also. Okay, so in that situation what happen, soil, if you see, the soil is also having the structure. If soil loses its structure, we call it erosion of a soil. Okay, त्यामुळे गरजेचं आहे की ते स्ट्रक्चर मेंटेन राहिलं पाहिजे, and to maintain the structure, soil required always required water. So that particular amount of water is not going to lose by a soil, and that is the thing. Soil only the water is only available for a plant is the except or the more water which is required for the maintenance of soil structure. Okay, जर maintenance of soil structure साठी लागणाऱ्या water पेक्षा जास्त water असेल तरच ते available आहे soil साठी, सॉरी plant साठी, नाहीतर soil plant ला water देत नाही. ओके, तर plant साठी जे water लागणार आहे तर ते वेगळी गोष्ट आहे, तर ते extra water द्यावं लागतं, soil स्वतःच water देत नाही. Nerve cell originated from, so it is originated from the ectoderm. So when you will have these options in examination and the one more option like all of these, so guys, only epithelial cell, epithelial tissues are synthesized from the all germ layers. ओके, बाकी सगळ्या सेलला लिमिटेशन्स आहेत तुमच्या टिश्यू वगैरे सगळ्या एपिथेलियल, फॉर एक्झाम्पल एक्टोडर्म पासून नर्व सेल बनलेले आहे, तुमच्या मेसोडर्म पासून काय बनल्यात, एंडोडर्म पासून काय बनल्यात, पण एपिथेलियल सेल तिन्ही पासून पण बनतात, एक्टोडर्म पासून पण, एंडोडर्म पासून पण, मेसोडर्म पासून पण. त्यामुळे ऑल ऑफ दिस वाला ऑप्शन असेल आणि वरती जर एपिथेलियल सेल असेल तर तुम्हाला ऑल ऑफ दिस वरती टिक करायचं आहे. नजल सेप्टम हॅज, नजल सेप्टम मीन्स द गॅप बिटवीन द नोट्स. ओके, सो फॉर एक्झाम्पल, okay, for example, this is notes, we know here is the gap. Okay, so here is I. So this question is about this material is made up of. So what is the answer guys? Nasal septum म्हणजे नाकाच्या मध्ये इथे जो पार्ट आहे ना असा जो which is actually the responsible for the making of nasal cavity or the मेकिंग ऑफ नजल कॅव्हिटी इंटू द टू. ओके. दोन नजल कॅव्हिटी मधला जो बॅरियर आहे, द बॅरियर बिटवीन नजल कॅव्हिटी इज कॉल्ड ॲज, the barrier between nasal cavity is called as, is there anyone? Okay, so it's a elastic cartilage, guys. Elastic cartilage is present here, which is responsible for the gap between, or you can say the separation between the nasal cavity. Okay, nasal cavity is divided into the two by the nasal septum, which is made up of the elastic cartilage. Which of the following is a nitrogenous polysaccharide? हा क्वेश्चन परत परत एक्झामिनेशन मध्ये येतोय मित्रांनो. नायट्रोजनस पॉलिसॅकॅराइड. तर आन्सर काय असेल रे? A polysaccharide. Poly means many or more than one. Saccharide means a sugar. So which of the following material is made up of the many sugar which is having the nitrogen content? Okay, so guys, the first answer we are getting from the Mansi. Mansi, I can't see your answer, beta. Can you send it one more time? Okay, good. It's a chitin. Okay, so the congratulations who got the answer. Chitin, it is a nitrogen containing polysaccharide. Okay, cellulose is made up of the glucose. Glycogen is made up of the glucose. Starch is made up of the glucose. Starch is present into the plant. Glycogen is the storage material into the animal. And the glucose, cellulose is present into the plant cell wall. Okay, it is a beta glucose, it is alpha glucose and the alpha glucose. मित्रांनो, अल्फा बीटा मधला फरक माहिती आहे का रे? अल्फा ग्लुकोज बीटा ग्लुकोज कळतो का? अल्फा ग्लुकोज आणि बीटा ग्लुकोज समजतो का? नसेल तर विचारा, मी एक्सप्लेन करून देतो. मला खाली कमेंट मध्ये टाका तुम्ही, जर तुम्हाला अल्फा ग्लुकोज बीटा ग्लुकोज मधला फरक पाहिजे असेल तर. Okay, me explain karun देईल. Your answer is chitin. Okay, sometime you will have question in examination like the heteropolysaccharide. Okay, so this is made up of the glucose, this is made up of the glucose, this is made up of the glucose. So your cellulose is a homopolysaccharide. Homopolysaccharide. Glycogen and the starch, these are also homopolysaccharides. Okay, हे सगळे homopolysaccharides एकाच प्रकारच्या शुगर पासून बनलेत. हेटेरोपॉलिसॅकॅराइड वेगवेगळ्या प्रकारच्या शुगर पासून बनतो. फॉर एक्झाम्पल तुमचा ब्लड ग्रुप, त्यानंतर हायलोरोनिक ऍसिड. आपल्याला हायलोरोनिक ऍसिड माहिती आहे की हायलोरोनिडेज नावाचा एन्झाइम अॅक्रोझोम मध्ये असतो, तिथं तुम्ही पाहिले. हायलोरोनिक ऍसिड इट इज ऑल्सो द हेटेरोपॉलिसॅकॅराइड. So bone dissolving cells are, if you see the bone is made up of the three type of cells. Okay, so which of the following is a bone dissolving cell? We know we have a thyroid gland responsible for the maintenance of calcium by TCT enzyme, thyrocalcitonin, and parathyroid gland. Okay, parathyroid gland. The enzyme is parathyrohormone, PTH. These two hormones are responsible for the maintenance of calcium in blood. Okay, so this is going to increase the calcium in a blood, and this is going to decrease the calcium in a blood. आज दोघं जण मिळून मेंटेन करतात. तर तुमची जी या क्वेश्चनचा आन्सर आहे, बोन डिझॉल्विंग सेल इज osteoclast which is responsible for the release of calcium from bone. So it is going to activate this cell, and it results into the uh, release of calcium into the blood. ब्लड मध्ये कॅल्शियम टाकून देते, त्यामुळे बोन छोटा छोटा होत चालतो, and that's why we called it a bone dissolving cell. Okay, so this is the correct answer. And osteoblast and osteoclast, if you see, the one is the inactive cell of the bone, and the second is a bone making cell. Okay, Harvesian canal is absent in. Harvesian canal म्हणजे काय मित्रांनो, पहा फॉर एक्झाम्पल हा बोन आहे. ओके, या बोन मध्ये जर तुम्ही पाहिलं तर काही पार्ट खूप हार्ड असतो. फॉर एक्झाम्पल हा पार्ट खूप हार्ड आहे. हार्ड असेल तर याला स्पेशल आपल्याला एक गोष्ट पाहिजे, म्हणजे यामधून ब्लड आणि नर्वस सिस्टम याचा सप्लाय न्यायचा आहे. ब्लड आणि नर्वस सिस्टमच्या सप्लायसाठी याला जागा लागणार आहे, आणि ती जी जागा आहे तिला हार्वेशियन कॅनल म्हणतो. इट इज रिस्पॉन्सिबल फॉर दी स्पेस, और इट मेक अवेबिलिटी ऑफ अ स्पेस फॉर दी ब्लड अँड नर्वस सिस्टम. नर्वस सिस्टम आणि ब्लड दोघांसाठी पण इथे स्पेस अवेलेबल करून देतात. जे स्पॉन्जी बोन आहेत, स्पॉन्जी बोन त्याच्यामध्ये अशी जाळी जाळी असते. फॉर एक्झाम्पल हा स्पॉन्जी बोन आहे, इथे यात जाळी असते, जाळीमधून जागा असते या दोन्हीसाठी पण, ब्लड पाईप साठी पण, नर्वस सिस्टम साठी पण. सो स्पॉन्जीस बोन डज नॉट रिक्वायर्ड दी absent of Haversian canal. Haversian canal, and the compact bone require or shows the presence of Haversian canal. Okay, so the correct answer is the spongy bone, which is showing absent of Haversian canal. Because there is a plenty of space available for the running of the blood and the nervous system, so they does not require special arrangement for the space. Okay, but in a compact bone there is a very low space available, and that's why they require special arrangement for the blood and the nervous system. Okay. Morula form at the end of the cleavage is called as is made up of the dash dash cell. Okay, so if you see what is meant by cleavage, cleavage is always looks like this. For example, this is your embryo, which is going to be a divide first from here, the second, third, and these divisions are called as the cleavages. Okay, so cleavage result into the first cell. For example, this is a embryo. First division, we have 2 cell. Okay, zygote, 2 cell. Second division, we have the 4 cell. 4 cell. Okay, third division, we have 8 cells. Fourth division, we have a 16 cell. Okay, and this is your morula. Morula is very good, beta. Absolutely correct. It's a 16 cell stage morula. After morula, we will have the blastula, then gastrula, then neurulation, and the other processes of the development. Okay, but the morula is present in a very a few animals like mammals. Okay, so other animals morula is not going to be considered. Scientists is having a doubt on a morula still, but still we considered it is the stage which is present in the development process, and it is 16 cell stage. Okay, 16 to 32. Some books will, I mean, you will have the some books like showing the 16 to the 32 stage. It's called as a morula. Okay, but 16 is the exact number we can keep in mind. Okay, uh, which is the following a correct order during the mitosis. Okay, so the correct order during the mitosis, we know the cell cycle. Okay, for example, this is a G1 phase, S phase, G2 phase, and the M phase. Okay, yes, very good, very good, very good, beta. Huh? Exactly, exactly, the answer is prophase. Okay, so the first phase is a prophase, second is the metaphase, third is the anaphase, and the last one is a telophase, and then we have a cytokinesis, and after cytokinesis we will have the two cells. Okay, so guys, when you read this, read like a PMAT. Okay, read like a PMAT. So it is the correct sequence: the prophase first, metaphase second, anaphase third, and the fourth one is a telophase. So it read it like a PMAT. The glycoprotein fertilizing is secreted by glycoprotein. Fertilizing नावाचं एक protein असते which is secreted by or which is present on, not secreted by, but the present on is the again more correct, present on. Kalyani, very good beta. It's present on ovum. Okay, and the sperm is having anti-fertilizing. Very good. No beta, Manse. The sperm is having anti-fertilizing and the ovum is having fertilizing. Required for species specific fertilization. Okay, and this event happens after acrosome reaction. Okay, this happens after acrosome reaction. Okay, so guys, are you getting my point? An international treaty known as a Montreal Protocol was signed to control the emission of. Okay, the Montreal Protocol कशासाठी आहे रे? To control the emission of UV rays, ozone, CFC, and oxygen. CFC. बर, exactly. To control the emission of CFC. Okay, and to protect the ozone layer. Okay, ozone layer ला protect करायसाठी CFC चं emission थांबवण्यासाठी याच्यामध्ये 27 वेगवेगळ्या कंट्रीज पार्टिसिपेट झालत्या आणि हा मॉन्ट्रियल प्रोटोकॉल आपण बनवला होता. ठीक आहे. Which of the following belongs to the class 5 of IUBP system of enzyme classification? So the system is OTLIL. जसं आता आपण पाहिलं PMAT तसाच आहे हा. OTLIL. ऑक्सिडोरिडक्टेज फर्स्ट आहे, ट्रान्सफेरेज सेकंड आहे, H हायड्रोलायजेस थर्ड आहे, लायगेजेस फोर्थ, आयसोमेरेज 5 आणि लायेज 6. ओके, तर तुम्हाला फिफ्थ विचारलाय. फिफ्थ जो आहे तुमचा तर तो आहे आयसोमेरेज. सो युअर करेक्ट आन्सर इज ए, जे तुम्ही ऑलरेडी पाठवले. व्हेरी गुड बेटा, इट इज करेक्ट ए. ओके, आयसोमेरेज. T is present at the second, so it is the second class. Ligase and lyases are the fourth and the sixth class. Okay, so it is enzyme commission which classify the enzyme as per the the functions. Okay, which of the following animal was selected by the Morgan for the study of a linkage? Apis indica is honeybee. Agrobacterium tumefaciens is the bacteria. Drosophila melanogaster in a banana. Okay, so not in banana, but it is actually your answer which is selected by the Morgan. Okay, and E. coli is again bacteria. Okay, it is an insect. Not Apis indica, but the correct answer is Drosophila melanogaster. By the Morgan, for the study of a linkage, we know the Morgan when working with this insect, the Drosophila melanogaster, it is having the few dominant insects, and he just try to understand why there is the a different type of insect like a red eye and the white eye, again the change in the structure of the wings, and after study he got that okay, each and every time when there is a white or the recessive color of a eye, so each and every time it's a male, and because of the observation he calculated that maybe the gene is present on X chromosome, and now the concept is come from there, that is the sex chromosomal or the sex linkage. Okay, sex chromosomal linkage which is coming after the experiment of Morgan. Okay, the human genome project was initiated in the human genome project is was initiated in 1990. Yeah, in 1990, and the plan to end it into the 2005, but it end very soon in 2003 before the 3 year, and uh, it complete the sequencing of human genome. Okay, so the project was the sequencing of human genome, sequencing of human genome to know the number of genes. So we have 30,000 of genes in our body. Okay, 30,000 genes आहेत आपल्या बॉडी मध्ये. If at the end of a meiosis the four daughter cells have the four chromosomes, how many chromosomes were in the mother cell? Okay, so the meiosis always started from the 2n, it is result into the n and the n, and again the meiosis 2 result into the n and the n. Meiosis 2 and the meiosis 1. Okay, so here the value of n is given here, the 4. Okay, so the n is equal to 4. Cell have the 4 chromosomes. Okay, how many chromosomes we are present in a mother cell? This is your mother cell. Okay, so very good guys, the correct answer is 4. Very good. Okay, because the n is, sorry, not n is 4 already given, and the 2n, so 2 multiplied by 4 is equal to 8, and the correct answer is very good, 8. Okay, the correct answer is 8, because what we got is a haploid cell which is coming from the diploid. So the chromosome number is always double in a diploid and half in a haploid. So the haploid word is coming from the actually haploidy level. How many NH3 are required for the urea? Okay, so the urea is synthesized into the liver in a Cori cycle. Okay, and it is the combination of the ammonia. It is made up of the ammonia and the carbon dioxide. Okay, so what is the answer, guys? So for urea, we required two ammonia, two NH3 with a one carbon dioxide. Okay, so the one carbon dioxide and two ammonia comes together and they form a urea, and which is filtered by the kidney and come out of the excretory system in the form of urine. Okay, secretion of renin from the Jg apparatus or the Jg cell is due to fall in. Okay, so the if you see the Jg cells, they are looks like this. This is Bowman capsule cup like a okay. So your Bowman capsule and this part is very close to each other, and this type of C GFR. Okay, so we have one answer, but it is not correct. Okay, so these are very close to each other, and when the glomerular blood flow, the glomerular the filtration pressure or the uh, blood pressure decreases, so it result into the uh, secretion of renin, and so the all of these is a correct answer. Okay, so the all of these is a correct answer, everything which is going to be a decrease in this situation, because it is happen because the volume of blood is going to be a decrease. Okay, so the due to the low volume, the pressure automatically decrease, and then renin start the conversion of angiotensinogen tensinogen to the angiotensin 1. This angiotensin 1 is converted into the angiotensin 2. So this conversion is by renin. Okay, this conversion by ACE in a lung. Okay, this angiotensin is coming from the liver. This is happen inside of blood, and after the lung, this angiotensin 2 enters into the uh, adrenal gland, and aldosterone is released, which result into the reabsorption. इथे जागा नाही वरती लिहितो मी reabsorption of sodium. ओके, कोणी रे काही शंका आहे का? असेल तर विचारत चला. ठीक आहे. Reabsorption of sodium and result into the reabsorption of water. Okay, reabsorption of water, and the volume increases. Volume of blood or the body fluid volume is going to be increases. अशी प्रोसेस आहे टोटल. याला बायोकेमिस्ट्री म्हणतो आपण मोस्टली किंवा हॉर्मोनल कंट्रोल आहे. ठीक आहे. अमाइनो ऍसिड प्रेझेंट इन हिस्टोन proteins are histones are the DNA binding protein. Actually hint DNA binding protein. Okay, so it is having the dominancy of amino acid. So guys, is there any answer? Arginine and histidine, arginine and lysine, arginine and cytosine, and lysine and the histidine. Okay, so the one thing is correct from the option. Arginine is present there. So the only we have to find the second one. The way we have one answer. The first one. Okay, we will discuss it definitely. So Kalyani, better histidine is not present there. Okay, so the first answer is not correct. Now you have two options. Cytosine is not amino acid. Okay, so the lysine is a correct answer. Arginine and lysine. Why arginine and lysine? Because it's a DNA binding protein. If you see the charges on a DNA, it's a negative charge due to the presence of a phosphorus. Okay, and these both proteins are the positive charge proteins. Okay, so we require positively charged protein which comes together and they form the histone proteins. Okay, the histone protein comes together, they form a nucleosome where the DNA is actually wrapped over. Okay, so arginine and lysine are the dominant amino acid present inside of the histone. Digestion of which of the following yields more energy? Okay, so which of the following is the largest source of energy? Carbohydrate, proteins, lipids, and the vitamins. Okay, so the first thing, vitamin is not a source of energy. Okay, so we are not going to use a vitamin as a source of energy. It's not a source of energy, so let it be. So we have only three options: carbohydrate, protein, and the lipids. So is there anyone which is having the answer of this question? Okay, A, carbohydrate. Okay, so we have one answer, carbohydrate. We will discuss is it correct or not. Okay, बाकी लोक काय करतायत, आठवतंय का काही? So actually the digestion of lipid yields more energy. Okay, then protein and then carbohydrate, because if you see the kilocalorie or the calorie energy in a calorie per gram, so you will have the highest value of a lipid, and that's why your answer is a lipid. Okay, carbohydrate is easiest way to make a energy, but the lipid is more energy yielding compound as compared to the carbohydrate. So the lipid is at the first position, protein at the second position, and carbohydrate at the last position. Okay, so thank you very much, guys. If you have any doubt, you can ask me in next session also. So this is Crack Maharashtra CET. If you do not have subscribe, first of all, subscribe this channel and hit on a bell icon. So, and when you are taking the subscription, do not forget to use this Abhijit as the subscription code. It will give you the 10% discount reduction in a fees. Okay, and this is Telegram channel for the MCQs and the notes. Okay, so thank you very much, guys. Thank you very much. We will see 7 p.m. today with a new session. So, okay, so thank you very much. Have a nice time. Bye bye.